Hello, good afternoon, everyone. Hope you have been having a good week so far, and good job on the quiz. The class average was substantially higher compared to the first quiz, so well done on that. And the lab reports, they were generally well done as well. So we will be doing lecture 5 today, and that's the last lecture that you're responsible for on test number 1. Okay, test 1 is going to happen after next week. There will be no class next week because of engagement week, okay, reading week. So you can take a bit of a break from school and just rest up a little bit. And when we return, we will actually have the class on Monday for the review. That's February 27th. And then the test is going to take place on March 1st. I will explain that again later on before you go when everybody shows up. But yeah, metabolism. That's this is a heavy lesson. It has a lot of information in it, and let's try to go through as much of it as we can. So the overall reaction for cellular respiration goes like this. We have glucose reacting with oxygen, and in the process, it becomes carbon dioxide. And water. And of course, the whole point of this is to create some ATP. And in terms of the chemical formula, it will be C6H12O6 plus 6O2, producing 6 carbon dioxide, 6 water molecules, and up to 36 ATP. Now, this is considered as a redox reaction. A redox reaction is a reaction that involves the transfer of electron. Transfer of electron. And in this case, the glucose is being oxidized. Okay, so oxidation means loss of electron, whereas reduction, that is gain of electron. Right, so in a redox reaction, something is losing electron and something else has to pick it up. So like I said, the glucose, we will be removing electrons from it, and at the end, when you remove the electrons from it, it will ultimately become carbon dioxide. Okay, so glucose is oxidized into carbon dioxide. Well, where do those electrons go? Well, they will be, I mean, they're going to go some places in between, but ultimately those electrons will end up being on the oxygen. And when the oxygen picks up the electron, they will become the water molecules. So they will be reduced into H2O. Okay, so along the way, this transfer of electron is going to allow us to harness the energy from the glucose and use those energy to produce ATP, the usable form of energy in a cell. So that's an overview of the reaction. Does anybody have any question about that? Okay. Now I'm just going to do an overview for you, you know, review if you have watched the recordings and and like a preview, I guess, if you haven't done so yet. So cellular respiration actually takes place in 4 major stages, 4 major steps. Okay, so the first stage is what we call the glycolysis. Followed by pyruvate, oxidation. And then we have the Krebs cycle. And lastly, we have the electron transport chain, or ETC for short. Okay, so that's the 4 major stages in cellular respiration. So let's take a look at where they happen. This is our cell, and I'm just going to draw a really big mitochondria here. It's not to scale. Okay, and I'm drawing the cristae right now. So glycolysis, the first step in cellular respiration, is going to take place here in the cytoplasm. And in a very simple sense, the glucose is going to split into 2 pyruvate. More on that later. The pyruvate will then enter the matrix of the mitochondria, and that's where step 2 and 3 will take place. Pyruvate oxidation and Krebs cycle will happen in the matrix. And finally, step 4 is going to be happening in the cristae right here, the foldings, right? I guess you cannot really see the 4. So there we go. Step 4 is going to happen in the cristae. I think I just made that worse. Hold on. There, step 4 happen in the cristae. So let's go back to our study guide here. Just write down the step first. So again, first step is glycolysis. Glyco means sugar. Lysis is to break. And that happens in a cytoplasm. Second step, pyruvate oxidation. What that means is we are going to rip more electrons from pyruvate. We're going to remove electron from pyruvate. And that happens in the matrix, the innermost portion of the mitochondria, right? And the third step is the Krebs cycle, which takes place also in the matrix. And lastly, we have the electron transport chain, or ETC, and that happens in the cristae, the foldings of the inner membrane, right? So that's an overview of all the steps and where you would expect them to happen. Do we have any question before moving on?
For the key product, what are you really like asking?
Sorry, come again.
For key products, what are you really asking?
Yeah, we're gonna fill that in together, okay? So let's take a closer look at what's actually going on in this process. In the first 3 stages, glycolysis, pyruvate oxidation, and Krebs cycle, what your body is focusing on is to harness the energy from the glucose. And that means we will be removing high energy electron during these 3 steps. Okay, so high energy electrons are released from the first three steps. Okay, and these high energy electrons, they will be captured by energy carriers. And these energy carriers will transport the electron to the ETC, where we will be using those electrons to make ATP. Okay, so high energy electrons are released. I'm just going to draw it like that, electrons. And then we have energy carriers. And there are two types of energy carriers that was mentioned in class. Does anybody want to tell me what are the names of those energy carriers? Energy carrier, electron carriers. We talked about two of them. Anybody know what they are? No? Okay, well, one of them is called NAD+, and the other one is called FAD. So these are like anti-taxis, and the electrons are like passengers. The NAD+ will pick up the electron, and afterwards it will become NADH. Similarly, FAD will pick up the electrons, and afterwards it will become FADH2. Don't worry about where the H comes from. Just, that's just what they're called. Okay. So that's the occupied version, and then this is like the empty. When they're empty, they can pick up electrons, and then they become full. And then what happens is these NADH and FADH2, they will travel to the electron transport chain, and they will let the electron come off, and it will cause them to become NAD+ again. Similarly, this is going to go to the ETC, and it's going to let the electron get off, and then it will become FAD again, right? So the NADH, the NAD+ and the FAD, once they let the electron off, they will go back to the first three steps and pick up more electrons, and then they will come here, and then just back and forth, kind of like a shuttling surface, okay, between, campus and nearest bus station around your home or something. So pick up passenger, drive them to the fourth step, let them off, and then go back and pick up more passengers. Does that make sense? Okay. So another thing that you should know is that each NADH contains enough energy to make 3 ATP, whereas each FADH2 contains enough energy to make 2 ATP. Okay, so they're not really the same, right? One has more potential energy in it compared to the other. So NADH is able to make more ATP compared to each FADH2. And I should also ask you over here, when the NAD+ becomes the NADH, how would we describe this? Would we say NAD+ is oxidized into NADH, or will we say NAD+ is reduced into NADH? What's the right way of describing that? Oxidized or reduced?
Oxidized.
Okay, that's a good try. But what does oxidation mean? To oxidize is to lose electron, right? Okay, oxidize means loss of electron. And over here,
Oh,
anything,
here's a gain.
yeah,
Oh, here's
it's
gained. Oh, okay.
right, exactly, right. So this is picking up electron. So this is reduced, reduced. Okay. And then when they get to the fourth step, then they are oxidized. Okay, because they let the electron off, right? So, again, any question before we move on? Please ask. I mean, if you have a question, it's very likely that someone else also has the same question in the class. Okay. Let's take a closer look at the glycolysis then. So glycolysis, like I said, glyco means sugar or glucose, and lysis is to break. So glycolysis literally means to break the glucose. Okay. So there are two phases here. There are actually 10 steps, right? But we don't have to memorize the 10 steps. We just have to know it's broken down into two stages, two phases, we'll say. We have the energy investing phase. Okay. So in this part of the glycolysis, what you do is you want to invest a little bit of energy. Okay? You give the glucose molecule a little bit of a boost of chemical energy, making it a little bit unstable, so it's going to be willing to react further. And to do that, we will be adding at 2 ATP to glucose. Okay, so this again energizes the molecule. It's more likely to react. That's the key purpose of the energy investing phase. And then we have the energy yielding phase. Yield is to produce, right? So this stage is where you will get some of your money back and hopefully more, right? It's like real life investment, right? You pay a little money, invest something in the bank and hopefully you get money back later on. So in this case, what we are getting back is we are actually getting 4 ATPs. And that's not it. You will also be getting some electrons that are released, and we will be capturing those electrons, right? And that will generate 2 NADH, like so. Okay, so in combination, this is more than what you have invested, right? You only invested 2 ATPs, and then now you get 4 back, right? So that means you have a net gain, net ATP equals to 2. You make 4, you take away the 2 that you invested, you actually, earn 2 ATPs. But this NADH, where will this NADH go? Please let me know. Can someone let me know what's the fate of this NADH? What's the destination? Where will it go? I can't tell if you guys don't know the answer or you just don't want to tell me. It's hard for me to judge without seeing the expression on your faces. So let me ask again, right? So the NAD plus picks up these electrons, high energy electrons from glycolysis, and it turns into NADH. Now, where does the NADH take those electrons? Okay, you can type the answer in the chat box if you don't want to talk, or you can private message me. Okay, Chisom say, I think we all don't know. All right. Maybe. So let's go back here. We were saying the first three steps, glycolysis, pyruvate oxidation, Krebs cycle, high energy electrons are released, right? Those high energy electrons, they are like little passengers. They get on the carrier, the NAD plus and FAD. These carriers becomes NADH and FADH2, and they will take them to the fourth step, the electron transport chain, where the electron will get off. And then the electrons will be used to make ATP in the last step. Okay, so once the electron get off, the energy carrier will become NAD plus and FAD again. And then they will go back to the first three steps, pick up more electrons, and then drive them to the ETC. And this just keeps on repeating itself. Is that better? Do you guys understand it more now that I've explained it a second time? And again, it's okay if you don't get it. You just have to ask me so I know what is it that you don't understand and I can explain it again. Just gonna pause for a moment and see if anyone wants to ask anything. Not to draw you back. Oh, that's okay. Yeah, but yeah, I mean, if you have questions, just feel free to ask. We can go back. It's not a problem at all. Okay. So are we okay now? Okay, with but could you explain cellular respiration again? Oh, okay. Cellular respiration is just this overall equation, right? Glucose plus oxygen produce carbon dioxide and water. This is the main reaction in the body that's responsible for making energy for us. Okay. And what we're saying is that this reaction doesn't happen in a single step. It's broken down into several stages. And the stages are listed here. Step 1, step 2, 3, and 4. Of the four stages here, the first 3 stages involves releasing high energy electrons. The energy carrier will capture those electrons and then use them in the 4th final step to make ATP. That's basically the relationship here. All right. Okay. And again, right, this whole thing is explained in greater detail in the lecture recordings. Okay. So the do this all take place in the mitochondria? Well, not really, right? We wrote it down here, right? Glycolysis actually happens in the cytoplasm. Okay. And then 2 and 3 happens in the matrix, and 4 happens in the cristae. Is that okay? All right. Let's see what's going on here. So overall, what happens is we started with glucose, and glucose has 6 carbons in it. And then the glucose will split into pyruvate. Which is 3 carbons each. Okay. So pyruvate will go on and be further metabolized, and in the end, it's going to have to be able to generate even more ATPs in step 2, step 3, and step 4. Okay. So that's basically the main reaction for glycolysis. So if you have to fill in the key products here, we would have 2 ATP and this is net, right? It's the 4 minus the 2 that you have invested. We're gonna have 2 NADH as well. And finally, we will have 2 pyruvate. Okay. Those are the key products. So let's do some poll questions. And you guys have to really participate to some of these poll questions, because otherwise I don't know if you are with me, right? Okay, so let me just give me a second. I gotta pull out the poll questions. Metabolism. Okay, so this is a simple question. Let's take a look.
The poll has blocked the question, so I cannot see the question.
A poll has blocked the question.
No, you
Here.
can.
You can move the poll window away, right? You know that? Like you can actually click on the poll and then drag it away. Like, do you see my poll here? Like on my screen, blocking the question? You do, right? You see it? Or no, you don't see it?
No, it doesn't.
No, okay,
You
but
just
like
have
you can
just have to
literally
move.
drag the poll window away, okay? Yeah, or minimize it, exactly. So I have 4 responses out of 5 now. Out of the 15 of you that's here, that's only 1/3 of you. Ooh, you guys are not making this easy for me. Okay, in any case, that's okay. I mean, we are all adults, and if you choose not to participate, that's okay as well. The cellular respiration, the molecule that is being oxidized is the glucose. Okay, the glucose will be oxidized into carbon dioxide, right? Okay, let's take a look at this next one here. And I mean, if you're not doing the polls on the screen, you can still write down the answer, right, for yourself, and then you can compare it to what I tell you. So NAD plus is blank into NADH when it accepts electron. We actually wrote this down earlier, right? Over here, NAD plus and the other energy carrier, they are reduced when they pick up electron. They're gaining electrons. That's why we call that reduction. So the answer is C. Okay. Let's see, we have other questions here. How many net ATPs are produced in glycolysis? Okay, very good. We have more participation this time, and most of you got it right. Correct answer is 2. This is asking for net production. It's not 4. I mean, you do produce 4, but then you have to take away the 2 that you've invested, right? So overall, you only really made 2. The answer is B. Okay, how about this one? Okay, the correct answer is 3 carbons. In the notes, we wrote that glucose has 6 carbons, and when you split it into 2 molecules, 6 divided by 2 gives you the 3 and the 3. Okay. And this last one here, let's do one more. All right. So a lot of people chose ATP. That's what happened to my other class too when I did it like in person with them. The correct answer is actually pyruvate, right? So I know why you choose ATP, right? You're like, oh, ATP, that's the energy molecule, right? And we generated some ATP, so of course it has the most chemical energy. But don't forget, in cellular respiration, we are making a total of 36 ATP. And in glycolysis, we have only made 2. Right? The pyruvate that we get will go on to step 2 and step 3, where you will be creating the rest of the 34 ATPs, right? So by the end of the reaction, most chemical energy is still locked in the pyruvate, okay? Not the ATP. So that's that. All right, we're gonna move on then to the next part. Let me just rewrite this for you so it'll be easier to follow. Let's put a break here. We started with the glucose, which has 6 carbon, and then it's gonna split into 2 pyruvate, right? Pyruvate, 3 carbon, pyruvate, 3 carbon. And over here, you invested minus 2 ATP, but in the end you get 4 ATP, and then you get 2 NADH, right? So now, where will the pyruvate go? This happens in the cytoplasm, right? Can someone please tell me where will the pyruvate go next? This is glycolysis, right? This is glycolysis, right? And then the next step, where is it going to take place?
Matrix.
In the matrix, that's right. Right, thank you. So this is going to go into the matrix. And in the matrix, we are going to have the second reaction, which is the pyruvate oxidation. Okay, and each pyruvate is going to undergo the same type of reaction. So there are 3 steps that are happening here. We will remove a carbon dioxide, And then we will extract more electrons from the pyruvate. So we will have a NAD plus that comes along and pick up the electrons to become NADH. Same thing over here. And no, it has to be a matrix, okay? Because there are different parts of the mitochondria, right? And different things actually happens in those different parts. So we want to be specific, okay? And finally, the molecule by now has, we have extracted quite a bit of electrons from it. We've made some ATPs from it. So by now, the molecule is a little bit running out of juice. Okay, it's running out of energy. So we want to recharge it a little bit. We want to re-energize it. And to do that, we are going to add something called coenzyme. Coenzyme A. So this is to recharge the molecule. Okay, so same thing over here. We're going to add coenzyme A. And at the end of the second step, we will have acetyl-CoA. All right. So this here, this whole thing, that is the pyruvate oxidation, which again happens in the matrix, whereas this happens in the cytoplasm. So tell me, how many carbon does the acetyl-CoA have? How many of the original carbon from the glucose do we have at this point? Well, the pyruvate had 3, right? And we removed carbon dioxide from it. So now it's going to be 2 carbons. Okay. So if we go fill in our chart here, the main product for pyruvate oxidation would be 2 carbon dioxide, 2 NADH, and 2 acetyl-CoA. And all these products are for each glucose, per glucose that you metabolize. All right. So next thing that happens is that we will have the acetyl-CoA. It's in the matrix right now, and in the matrix there is another molecule called oxaloacetate. And this one has 4 carbons in it. So the 4-carbon oxaloacetate will combine with the acetyl-CoA, and that's going to form citrate, which has 6 carbons in it. Okay. And then a bunch of things happen, which we don't really have to know. What we do have to know is that this is going to release 2 carbon dioxide, right? So if you take the 6 minus the 2 carbon that we released, it will go back and become 4 again. And it will be available to combine with another 2 carbon, and then it just keeps on happening. Okay. Also, over here, we are going to generate 3 NADH and 1 FADH2. Okay. But don't forget, this happens twice, right? So this acetyl-CoA will also be undergoing the same thing. I mean, we can draw it again. It'll combine with this 2 carbon, and then it will become citrate. 6 carbon. And then it will be releasing 2 carbon dioxide, 3 NADH, and 1 FADH2. Okay. And also, we will be making 1 ATP. Okay, so you have to remember the main products. And all this happens in the matrix. So this here is the Krebs cycle. Happens in the matrix as well. So let's write them down. For the Krebs cycle, we will produce a total of 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. And again, this is for every glucose that we break down. So you can see by the end of Krebs cycle, the glucose is completely broken down into CO2, right? We can account for all 6 carbon dioxide. 2 plus 4 is 6, right? So we started with 6 carbon in the glucose, and now they're all released as carbon dioxide. Okay. Again, I ask you, any questions you have for me? Do you want me to clarify anything before we move on? All right. So let's switch gear and focus on these ATPs. Let me write down for you. ATPs produced in glycolysis and Krebs cycle. We don't make any in the second step, right, in pyruvate oxidation. So ATPs produced in glycolysis and Krebs cycle uses a method called substrate level phosphorylation, okay, or SLP if you want to write short. So substrate level phosphorylation needs 3 things. We have an enzyme. You know what? I'm just gonna copy it from the PowerPoint rather than drawing it right here. Okay, so it requires 3 things. Okay? We need an enzyme to help facilitate catalyze a reaction. Over here, we have a donor molecule that is willing to donate a phosphate. So this one has 2 phosphate. It doesn't mind giving up one of them. And then we need to have an ADP. Okay, so phosphorylation. To phosphorylate something is to add a phosphate to it. So the enzyme will take the phosphate from the donor, transfer it to ADP, and that's going to make ATP. Okay? So the ATP that we made from glycolysis, the 4 of them, or net of 2 ATP, as well as the 2 that we make from Krebs cycle, uses this method. Okay? So only about 11% of ATPs are produced this way. Okay. So like 4 of them, right, out of the 36. Those are the ones that are produced by this particular method. So in a chart here, we have ATP, ATP here. Right? Put a star here. These are produced by substrate level phosphorylation, the process that I just explained to you, right? With the enzyme, with the donor, and the ADP. All right, so let's pause for a moment. Let's recap what we know so far. We know glycolysis, we know cellular respiration is separated into all these steps, right? 1, 2, 3. And I've been telling you, during these 3 steps, we are removing electrons from the molecules, from glucose, and those electrons are full of energy. So we want to capture them. Okay, so you have 2 ways of capturing it. You can either use an NAD plus or an FAD, and you capture all these electrons and you take them to the ETC, the last step. Okay, and in the last step, I'll show you what happens to those electrons. Right, so along the way, that's the primary purpose, right? In glycolysis, you create 2 NADH, pyruvate oxidation, and additional 2 NADH. And then for Krebs cycle, you get 6 NADH and 2 FADH2. In addition to capturing high energy electrons, we were able to make a little bit of ATP along the way, right? Not a huge amount, but we get 2 net ATP from glycolysis. We have 2 net ATP from Krebs cycle, and together that's like 4. It's not a lot, but it's better than nothing. And those ATPs are produced using a method called substrate level phosphorylation, where an enzyme will take a phosphate from a donor and put it onto ADP to create ATP for us. So only a small percentage of all ATPs are produced this way. So that's the story so far. Are we together on this? Okay. So I realize it's a challenging lesson. So please let me know if I can make it any easier for you. Okay, let's do some polls and see where we stand. Let's try this one. All right, any last attempts before I close the poll? All right, let's take a closer look. NAD plus has more chemical energy than NADH. That is not correct. because NAD plus is before they pick up the high energy electron, right? So it's more energetic after it picks up the electron, less. NADH has more chemical energy than FADH2. That is correct. Each NADH has enough energy to make 3 ATP. FADH2 can only make 2 ATP. So this is correct. NAD plus can be reduced into NADH when it picks up electron. And this one is not true either, because we need the NAD plus to pick up those electrons that are being released in glycolysis. If NAD plus is not around, nothing is going to be picking up those electrons, and glycolysis is just going to be stuck, and nothing's going to be happening. Okay. This is a important question. You should try this. Okay, it has been a minute, and I have a few attempts here. And so to do this, we just have to do some math. 4 NADH here, so each NADH can make 3 ATPs, right? So we multiply this by 3, that gives me 12. And then this one, we're going to multiply by 2, because each FADH2 can make 2 ATP, so 2 times 3 gives you the 6. And we just have to add them together. 12 plus 6 is 18. All right. Any question? It's pretty straightforward. Okay, this one is harder. I had a class of 30 yesterday, like in person, and nobody got this right. Let's see if you guys can do a little bit better. And so over here, we have to take a look at 1 turn of Krebs cycle. 1 turn. If you look at the notes we wrote earlier, with the circle, right? It produces for each turn, okay, it produces 3 NADH, 1 FADH2, and also 1 ATP, right? So this one will go on to step 4 and make 3 ATP each. So that's going to be 9 ATPs. This one will go on to step 4 and make 2 ATPs. and that would be 2 ATPs in the end. And then this one is just 1 ATP. If we add up all these things, we get 12 ATP for one cycle. And this is talking about 5 turns. So 12 times 5 gives me 60. Okay, so that's how you get the answer. Okay, moving on. Let's go to the electron transport chain. Let's take a look at what happens there. I already explained this in class, but judging from the response here, I think it's a good idea to do some refresher. Okay, so this is the mitochondria. This is the inner membrane. And what we're gonna do is we're gonna take a slice of it. Okay, and then we're gonna zoom in. So here we have the outer membrane. And then we have the inner membrane. And it forms these foldings to increase surface area, called cristae. The space between the outer membrane and the inner membrane, that is the intermembrane space. We have the matrix on the inside. So the electron transport chains, they are basically a series of membrane proteins. So that's the ETC there, electron transport chain. And you have many of them. Okay, you might have another one here, and then maybe another one here. And they're just a group of membrane proteins that are embedded in the inner membrane. And so all those NADH that we've generated in the first 3 steps, all those FADH2 that we've generated, they're going to come to the ETC. Okay, so NADH will deposit the electron in the first protein. And of course, after it deposits the electron, it will become NAD+ again. All right. So where will this NAD+ go now? Anybody remember? Where is it going to go?
You mean the NAD?
NAD+, that's right. NAD right here. Where is it
Go
going to go?
back to ET. Go back to ETC.
But it just went to the ETC, right? The NADH put the electron down at the ETC, and now it becomes NAD+ again. Where will this go?
Oh.
So this one will go pick up more electrons, right? From Krebs cycle, maybe.
Oh.
Right. So remember the first 3 steps, right? The electrons keep on getting released, and we need to capture them. Okay. So now that we release the electron onto the first protein, the electron will get passed down the ETC. And as the electron passes down the ETC, the energy from the electron will be used to pump H+ from the matrix into the intermembrane space. Okay, and same thing's going to be happening here. It's going to be pumping H+, pumping H+. And over time, you will have a lot of H+ in the intermembrane space. We will have some here, but not as many. So that creates a high concentration in the intermembrane space, and then we will have a low concentration of H+ here. All right. Now the H+, they really want to come back in because things naturally wants to go from an area of high concentration to an area of low concentration. So they want to come back in, but they cannot because the membrane will block them, right? So you build up this reservoir of H+, kind of like a water dam, right? You build up all this water, and when you let the water flow through a turbine, it generates electricity. So similarly, there is a special protein that is in the inner membrane, and they usually draw like that. Looks like a little doorknob. And this protein is called ATPase, or ATP synthase. Same thing, depending on where you're reading it from. And this allows the H+ to pass through. The H+, these H+ is going to go through the ATPase, go back into the matrix. As this is happening, as the H+ goes through the ATPase, the power from the gradient will be used to add phosphate onto ADP. And that's going to be generating ATP. Okay. So ATP that are generated this way, as opposed to this way, this is called oxidative phosphorylation. Okay, so a majority of the ATPs are created this way. We are adding phosphate onto ADP through oxidative reaction, right? And oxidative reaction means we were removing electrons from the glucose, right? Do we have any question about what I've just said? There are a couple other things I want to mention, but first, questions. Okay. So secondly, in addition to the NADH, we also have the FAD H2 that we haven't talked about, right? So the FADH2 will do the same. FADH2 will do the same thing. Just choose a different color here. So we have the FADH2, and then it will become FAD. The only difference is the FADH2 will deposit the electron at a lower point on the ETC, not the first protein, but the second protein. Okay, so it's not going to be able to pump as many H+ over, and therefore it's only capable of creating 2 ATPs as opposed to 3 ATPs for NADH. Okay. Now there's one last thing that we have to talk about. Okay, let's take a look at the ETC again. I'm just going to draw them separately here. Okay, right here. Each protein, we call this protein 1, 2, and 3. Each protein in this in the series is going to be having a higher attraction for electrons compared to the previous one. I see hands up. Yeah, go ahead, please.
Sorry, it's by mistake.
Oh, no problem. So each subsequent protein in the ETC will have a higher attraction for electron. So when the electron gets on the first one, it will get passed on to the next one and then to the next one. Okay, but at the end, something has to pull the electron away. Okay, otherwise it's going to stay there, and then the next one will not be able to come. It's like, you know, at the park. Sometimes I take my kids to the park, and then you know they would go on and then slide down, and then they would want to go again. But then there is some kid at the bottom of the slide that just stays there. It's not like they're not going away. So then the next person cannot slide down, right? And then people get frustrated. And so what happens is the parent has to come and then take the kid away, and then the next one can go. The electron is like the kid, okay? Slides down the ETC, it goes to the bottom. If it doesn't go away, then the next electron cannot go, okay? So who's the parent? What's gonna come and pick up that electron? Can anyone answer that question? I already told you the answer like way early on today in the lesson. Okay, what's gonna be picking up? The NAD was responsible for picking up the electron along the way, right? From step 1, step 2, and step 3. But ultimately, where will the electron go? Right? Think about the overall reaction. Ultimately, the electron started on the glucose, but eventually it will go to where?
ATP?
Good try, not quite. It will be oxygen. Okay, oxygen.
Oxygen.
Over here, you can see it, right? Oxygen. It's going to be reduced. Okay, and it's going to be reduced into H2O. So the oxygen is going to pick up the electron, and then it's going to turn into water. Oxygen is called the final electron acceptor. Final electron acceptor. Okay, it has the highest electronegativity. Okay, and this is a big word, but all it really means is like attraction. For electron. Okay. So number 2 has higher electronegativity than number 1. Number 3 has higher electronegativity than number 2. But oxygen has the highest electronegativity. It has the highest attraction for electron. That's why it's able to pull the electron off the chain so that the next electron can slide down. If we don't have enough oxygen, then the ETC is going to be clogged with electron. Okay? Without oxygen, the electron does not get off the ETC, then the next one cannot slide. If the next electron cannot slide, we will not be able to pump H+ into the intermembrane space. Without H+ here, they will not be going through the ATPase. And then we stop making ATP. That's why we need to breathe continuously. The oxygen that we breathe in allows this process to continue, and we need to have a continuous supply of ATP, without which we would die, right? Okay, so that is the electron transport chain. So we'll skip fermentation for now, and let's take a look at this chart right here. Okay, so let's try to calculate the number of ATPs that we are making, along the way and in total. So let's go back to glycolysis for a second. That is the first step in cellular respiration. How many net ATP did we produce in glycolysis? Does anybody recall? We wrote it down a couple times now, right? You should know, and we did the poll question.
2, net 2.
That's right. So through substrate level phosphorylation, with the help of an enzyme, we're going to be producing 2 ATP. And again, this is net. In terms of energy carrier, we have 2 NADH. Okay. Now, these NADH are actually produced in cytoplasm. That's where glycolysis takes place. So let's go back to our diagram here. Okay, this is the mitochondria, and this is the cytoplasm. So from glycolysis, you created these NADH. But it turns out NADH is really big. It's too big to go into the matrix. And so what happens is instead of having the NADH go into the matrix, it's just gonna pass the electron along. Okay, and then the electron will go through and a pair of FAD will pick them up. Okay, so again, just to say that one more time. These NADH are generated in glycolysis, which happens in the cytoplasm. The NADH generated there is actually too big. It won't be able to go into the matrix. So instead of the molecule, the carrier going through, they would just pass the electron along. Okay, so someone else has to capture that electron, right? So an FAD come capture that electron, and then you get FADH2. And then from there, it could go to the ETC. Right, so there's that extra step here. Because of that extra step, right, instead of producing 3 ATP each, okay, these are transferred to 2 FADH2, right, in matrix. These are cytoplasm. The number of ATP produced is actually just 2 times 2, which is 4 ATP. Okay, so 2 ATP from substrate level phosphorylation plus the 4 ATP produced here, that's a total of 6 ATP. Pyruvate oxidation, 3 things happen there, right? We remove carbon dioxide, we produce NADH, and we energize the molecule with coenzyme A. So actually there's no ATP produced directly from substrate level phosphorylation. We do have 2 NADH though, but this time they are already in the matrix. Okay, so they could go directly to the ETC. We times 3, that gives us 6 ATP. In the Krebs cycle, we have 2 ATPs from substrate level phosphorylation, 6 NADH in the matrix, 2 FADH2. We roll all these down in the notes and the table above. You can check the numbers later on. Times 3 here, times 2. So that will give me 18 plus 4 ATP. Okay. So that's going to be 18 plus 4 plus 2, which gives us 24 ATP. And if we add them up, 6 plus 6 plus 24, that's how we get the total of 36 ATP from metabolizing 1 glucose. Okay. That's that. Let's take a moment and look at the, I mean, you can do this on your own and check the answers later, but let's take a look at the fermentation here. Okay, there's a chart here. Let's go through it together. So everything that we learned so far, the Krebs cycle, pyruvate oxidation, that happens when we have an adequate supply of oxygen. Okay, but sometimes if we don't have enough oxygen, then we switch to fermentation. Okay, so there are 2 different types of fermentation depending on the organism that we're talking about and the conditions that the cell is in. Right? So here we have lactic acid fermentation. So that's something that could happen in a human cell. Okay, so muscle cell. When does it take place? This happens when oxygen demand, when the need for oxygen exceeds the oxygen supply. Okay. an example of when that could happen is, imagine you're running at full speed. Right? You cannot keep that up forever. If you run as fast as you can, maybe after 2 or 3 minutes, then slowly the oxygen that are going to your leg muscles is not going to be able to keep up with the demand in those leg muscles. Okay. So when the demand exceeds supply, you don't have enough oxygen. Not having enough oxygen will cause your ETC to clog up and your ATP production starts to slow down. So during that time, your body switches to lactic acid fermentation. And that happens in the cytoplasm. The final product is 2-lactic acid. And you will get NAD+. I'll explain the significance of this in a bit. The other type of fermentation is called alcoholic fermentation, and that happens in bacteria and yeast. Okay, and for them, this is just a norm. Okay, they don't have the cellular machinery to do Krebs cycle or ETC, so they rely on alcoholic fermentation. So almost always. And this also takes place in the cytoplasm. And what you generate is ethanol, carbon dioxide, and also some NAD+. Okay, 2 of each. That's why people can use bacteria to make alcohol, right? The ethanol that is generated is the drinking alcohol. Not all alcohols can be consumed, right? Like rubbing alcohol, that's isopropanol. It's gonna be poisonous if you drink that, right? Methanol, that's rubbing alcohol, also not consumable. And the carbon dioxide that you get from the yeast, that's how we make bread, right? That's what causes the bread to rise, to leaven. So let's take a closer look at these processes in this diagram. Okay, so we have glycolysis here, nothing new. I'm just gonna write glycolysis. And we already know in glycolysis, amongst other things, you're gonna be able to take the ADP and create 2 net ATP. And at the end, you're gonna get 2 pyruvate molecule, right? That's the pyruvate over here. So for bacteria and for yeast, like I said, they do not have the machinery to do Krebs cycle and ETC. So the pyruvate is gonna go on and become something called acetaldehyde. And in the process, you remove carbon dioxide. And again, that's what causes the bread to rise if you use yeast to make them. And then the acetaldehyde is going to become ethanol. Now, the names are not so important. What's more important is to understand the purpose of this. So glycolysis will release high energy electron. And as we mentioned multiple times already, we will need to have the NAD+ to pick up those electrons. So NAD+ will pick up the electron and then it will become NADH. Okay, but where is it gonna go? Well, there's no ETC, right, to go to. So eventually, if we don't have a destination for it, then you will use up all the empty taxis, so to speak. You will use up all the empty NAD+, and you will just end up have a bunch of NADH that has nowhere to go. And when you run out of NAD+, glycolysis cannot happen, right? So this reaction here, the pyruvate becoming acetaldehyde and then becoming ethanol, that basically provides an alternate destination for those electrons. And you are able to regenerate NAD+ in the end so that glycolysis can keep on happening. Okay, so fermentation here provides, and same thing for the other one, okay, provides an alternate alternate destination for NADH to deposit the electron, so that you can free up the NAD plus and let glycolysis continue. Okay. Similar situation here with lactic acid fermentation. We have glycolysis at the beginning once again. Right. The ATP will create 2 ATP. And in the process, we also will pick up the the electron with NAD plus and generating NADH. Oops, you can't really see that, can you? We go 2 NAD plus and 2 NADH. So in this case, the glucose also becomes 2-pyruvate. And instead of generating ethanol, because that's actually poisonous to our cell, we will create lactic acid. You might have heard of people say, you feel the burn from your workout the next day. That can be attributed to the buildup of lactic acid in your muscle. All right. Okay, so I encourage you to finish the rest of the study guide. I mean, I could fill it in for you, but it's probably much better that you fill it in yourself and then look up the answers to learn about it. And so no class next week, and the next time we will actually have the lesson on Monday instead of the Wednesday. Okay, give me a second here. Okay, the Monday is February 27th. Okay, we'll have a 12:30 class on Monday. That's going to be a test 1 review. I'll post the review package and you can work on it and stuff like that. But then we will take it up on Monday. And following that, the Wednesday, you will actually take the test. Okay, so the test, it's available from 6 to 11:59 p.m. And that would be the end of the first half of the course. Okay. If you need extra help, feel free to message me. We will, book a time to meet over Zoom during engagement week if that's something that you need. You can always just email me the question and I'll get back to you as well. All right, thank you for coming and have a good break. I'll see you all on the 27th. Bye for now.